Hello, my name is Jonathan Lynch. The title of my presentation today is Roots for the 10 Billion, Modeling Phenes and Phenotypes for Soil Resource Capture. OpenSimRoot is a dynamic, functional, structural model of root-soil interactions with the goal of understanding the relationship of root phenotypes and soil resource capture. It's been in development for nearly 30 years, involving extensive, unique empirical parameterization. A number of species can be simulated in OpenSimRoot. It's been open source since 2017 with 3 development nodes: University of Nottingham, Forschungszentrum and Penn State University. OpenSimRoot captures the biophysics of the dynamics of water, carbon, and nutrient movement in the soil, plant, atmosphere continuum. As a functional structural model, it also is spatial temporally explicit and precise in how specific root phenotypes interact with specific soil volumes. As an example, this image displays nitrate uptake kinetics in maize root systems as represented by OpenSimRoot, and this is based on empirical parameterization. You can see on the left Imax, that's the maximum velocity of nitrate uptake. On the right, Km, which is the affinity of the transporters for nitrate. Both of these parameters vary substantially within maize root systems. They also vary substantially in time. And this spatial temporal dynamism and complexity is captured by OpenSimRoot. This type of information is used to drive simulations such as this one. This shows 3 maize plants growing in the spring in a Pennsylvania soil. The colors represent soil water nitrate concentration. You can see that that's dynamic, spatial temporally dynamic because of movement of water as well as acquisition by the crop roots. OpenSimRoot has been critically useful in identifying avenues to breed more nutrient efficient crops, in evaluating trait utility, in discovering new traits, in estimating processes that we cannot control or measure, in evaluating phenotypes and environments that do not exist in nature, and in exploring decision spaces that are too vast to explore empirically. In the example I'll show today, which is how integrated root phenotypes can affect low nitrogen tolerance in rice, 2 of these capabilities are featured: evaluating phenotypes in environments that do not exist in nature and exploring very large decision spaces. So to parameterize OpenSimRoot for rice, we used shovelomics methods, in other words, excavation of root crowns in the field, monoliths, soil coring, both of which give us information about root distribution in the field, baskets, which allow us to capture detailed information about crown root phenotypes, rhizoboxes, which allow us to measure elongation rates of different root classes, and nutrient solution techniques, which allow us to measure physiological parameters such as root respiration in distinct root classes. As phenotyping platforms, we also availed ourselves of digital imaging of root traits for root architecture measurements, WinRhizo for root length measurements, and laser ablation tomography for root anatomical phenotyping. This is the result. This is OpenSimRoot rice for IR64, our reference phenotype. By 40 days after germination, SimRoot is simulating the growth of nearly 3 million individual root segments interacting with each other and with the soil around them. This image shows how some of those phenotypes manifest. On the top, IR64, our reference phenotype, showing angle, lateral root branching density of both L-type and S-type roots, and the number of nodal roots. Below those images, you can see variance we generated by changing the input parameters of the model, therefore allowing us to study the effects of individual changes on overall plant performance. This then is the result. This is OpenSimRoot rice in a low nitrogen environment. Once again, the color represents soil water nitrate concentration. In this simulation, by 30 days after germination, there were still gaps in the soil in this relatively dry scenario, which this model, by the way, includes soil mineralization, where there's nitrate available in the topsoil that has not yet been acquired by this phenotype, and nitrogen available in the subsoil. OSR rice accurately predicts the observed growth of IR64 under optimal and low nitrogen supply. In this axis, you see 30 days of growth. On the y-axis, shoot dry weight. The symbols represent empirical measurements of IR64 in the field, and the lines represent SimRoot predictions. You can see good relationship between empirical data and simulated data. And SimRoot tells us that different agroecologies will manifest as different nitrogen dynamics. In this case, we're looking at a range of nitrogen supplies. We're looking at simulated shoot dry weight at 30 days after germination as a function of whether we have dry season or wet season and whether we have non-puddled or puddled soils, and all of these affect performance at low nitrogen availability. Now, perhaps the simplest application of a model like this is to vary one phene, one trait, to see how it affects performance, and that's what we show here. For example, nodal root growth angle, or the percent of nodal roots with small diameters on the main stem, or lateral root branching density, et cetera, in 4 nitrogen regimes, and how they affect simulated shoot dry weight. And OpenSimRoot tells us there are some phenes, such as nodal root growth angle, that have a large effect on performance under low nitrogen. Others have a smaller effect. But this sort of a study doesn't take full advantage of the capabilities of OpenSimRoot, which can simulate many different phenotypes in many different environments. So we decided to vary many things concurrently to see which integrated phenotypes are best or optimal for performance in low nitrogen soils. In this case, we have 5 phenes that we feel to be important based on previous research, each of which is varied in 4 states, one of which, shown in red, is the IR64 reference phenotype as measured empirically. So the combination of 5 phenes in 4 states generates 1,024 combinations or integrated phenotypes, each of which is simulated under low and high nitrogen availability. So we have a total in this case of 2,048 simulations. And those simulations show substantial synergism among root phenes for improved tolerance to low nitrogen. In this figure, I'm showing 10 phenotypes, 1 to 10, and we're graphing those as a function of percent reduction in shoot biomass under low nitrogen. So the lower the number, the better the plant performance. If we estimate the expected contribution of each phene state to plant performance just through additive effects, we have these bars in red. And there's not much difference among the phenotypes if we look at their additive contributions, just adding up the contribution of each phene state, such as having higher branching density or having shallower growth angles. However, SimRoot is able to capture the interactions among these phene states through competition for internal plant resources, such as carbohydrate or nitrogen, competition for external plant resources, such as water or nitrogen, and trade-offs. For example, if you grow shallow roots, you have fewer resources to grow deeper roots, et cetera. And those interactions are quite significant. You can see in these 5 phenotypes, numbers 1 to 5, the simulations predicted a much better performance under low nitrogen when synergism was considered than you would predict from just the additive contributions. On the other hand, these other 5 phenotypes, 6 to 10, perform much worse in silico than when synergism was considered than when we just look at their additive contributions. So these interactions among phene states are very important in determining plant fitness. To estimate how these changes in plant growth and during vegetative phase could impact final yield, we linked OpenSimRoot to ORISA version 3. ORISA version 3 is an ecophysiological crop model simulation for rice that is a biophysical model that captures carbon and nitrogen and water dynamics and photosynthesis and so on. But it's not a functional structural model, it's just a process model. So we hooked these two things together. OSR rice then would simulate growth up to 30, 40 days, pass those parameters for vegetative development, leaf area index, nitrogen content, biomass distribution, et cetera, onto ORISA version 3, which then took those parameters and stimulated final yield in specific environments. This is a result of an example of one of those simulations. So this is a result of the integration of OpenSimRoot rice with ORISA, where we have the model has identified several phenotypes shown here that are predicted to be superior to IR64. And ORISA is able to simulate yield from those phenotypes. So here we have the range of phenotypes compared to IR64, their simulated yield in the year 2016, the actual precipitation of the year 2016, as experienced at Erie headquarters in the Philippines. And according to these simulations, on average, these phenotypes are able to increase yield by 14%, with the best performing phenotype 37% better than the yield of IR64. Now you'll notice that phenotype F actually performs worse than IR64 in this simulation, contrary to OpenSimRoot predictions. And we think that's because of this particular year, 2016, was a rather dry year. The nitrogen distribution in the soil was rather different than usual. But in this particular year, this phenotype was worse than IR64. And we're now conducting simulations such as this in multiple years and multiple sites to have a more robust determination of the effect of root phenotypes on plant performance under low nitrogen. So to summarize this OSR rice example, multiple integrated root phenotypes can improve rice performance under low nitrogen in silico. The utility of root phenes for nitrogen capture depends on the agroecology. In direct seeded, dry season agroecologies, such as I presented here, beneficial phene states are shallow nodal root growth angles, greater proportion of small diameter nodal roots, greater lateral root branching. And all of these things are under genetic control and can be selected in crop breeding. The utilities of nodal root number and the lateral root branching density of fine roots are dependent on the background phenotype. And finally, phene synergism is important in overall plant fitness. Conclude with a few comments on functional structural modeling. Functional structural modeling is useful in identifying crop breeding targets. The complexity of the fitness landscape for root phenes, including interactions with the environment, management, and the background phenotype, exceed the capabilities of empirical research. Let me point out that obviously empirical research is needed to validate these predictions, but empirical research is slow and expensive and can't do some of the things we need to do. So the modeling is useful in guiding our empirical research to the most fruitful avenues. The ability to generate novel phenotypes and project them to future environments exceed the capabilities of empirical research. Expanding compute resources and ongoing efforts to network multiple models across spatial temporal scales are increasing the power and utility of robust in silico approaches. Thank you very much. And in this slide, I show some of the participants who did all the work that I'm showing you today. Thank you very much.